Okay, good morning, everybody. Welcome to the morning session. We are scheduled to go from 9:30 until 12:15. Each speaker will have 30 minutes to do their presentation and also to handle questions. So some speakers perhaps may want to handle questions in the middle, but generally, if you can put your questions in the Q&A, we can get them to the speaker within the 30 minutes, the last 5 minutes or so. And then there's also a general discussion between 12 and 12:15. So that's pretty much how we're gonna function on that account. So we have five speakers. Our first speaker is Brian W. Kunkle from the University of Miami, the Miller School of Medicine. Dr. Kunkle is a research assistant professor at the University of Miami, Miller School of Medicine, the Department of Human Genetics, and the Hussman Institute for Human Genomics, where his work focuses on discovering genomic risk factors for Alzheimer's disease and related dementias through the application of genetic epidemiology techniques. The title of his presentation is Diversity, Disparity, Genetics, and Their Implications for Precision Medicine in Alzheimer's Disease. Dr. Kunkle, we welcome you.
Thank you. Morning, everyone. Thanks to the organizers for having me. I realized after I dressed this morning that I probably picked the wrong color shirt. so I hope you don't hold that against me. Yes, so let's see. Do I use the keyboard to advance?
Yes.
Yeah, okay. So just a brief overview of the talk. We'll talk a little bit about why we study the genetics of common disease such as Alzheimer's. Talk about two areas of research that I'm focused in, looking at genetic disparities in Alzheimer's and genetics, some of the differences that we see in race, ethnicity, and ancestry, and also sex in genetic underpinnings of Alzheimer's. We'll talk a little bit about what we've learned in terms of differences and similarities in genetics between ancestral groups and between males and females. We'll then talk about why these genetic ancestry and sex differences are important for precision medicine, especially, and for preventative strategies. And finally, we'll talk about what we need to move forward towards treatments and preventative strategies that work for everyone. So why do we study the genetics of common diseases such as Alzheimer's? Well, I think there are a few major reasons. The rationale for human genetics research in general is that by identifying genes with variants that cause a disease, alter risk, or prevent a disease, we can gain information that allows us to determine therapeutic and preventative strategies. For instance, we hope to identify new drug targets from this research by identifying genes that alter the proteins and ultimately the mechanisms and pathways that lead to disease. And we know that genetic targets are twice as likely to be successful in drug trials, showing the power of human genetic studies. So when part of this is when we identify a gene where a mutation alters the function or increase in expression. These are experiments of nature that are directly in humans, and we should be able to, use this information in pharmacological trials to be able to influence disease risk. Another major reason to study human genetics is prediction, where if we can gain enough information about the genetic underpinnings of a disease that has a large genetic component, we should be able to increase our ability to determine who's more likely to develop Alzheimer's or other complex disease. And we'll talk a little bit later about how this, what this predictive information can do for us. So how much of Alzheimer's risk is genetic? Well, estimates from family studies have it anywhere between 60 to 80% of late onset Alzheimer's is genetic, with the rest being social and environmental risk. The rare earlier onset form, of course, does have single highly penetrant gene variants, however, and less contribution from the environment. But for this talk, we'll really focus on the later onset, more complex form of the disease. What we're really trying to do ultimately with our studies. It's not.
Sorry, yeah, you can.
Yeah, it wasn't advanced. Sorry. So what we're really trying to do ultimately with our studies is fully describe the genomic underpinnings of Alzheimer's disease. It's also important to recognize that markers of drug response and adverse drug reactions vary in frequency within and across ancestry groups and between the sexes. And as I will cover in this talk, there's a lack of diversity in our research on the genetics of Alzheimer's, and also a lack of information on how the genetics of disease differ between the sexes. And this lack of information can contribute to disparities we see in Alzheimer's, and could also lead to disparities in our treatment and preventative strategies. So I really see three major areas of disparities: race, ethnicity, sex, and geography. And the two where genetics research, including research in my group, has shown there are differences in population groups and between males and females. Let's talk briefly about these disparities. For one, we know that Hispanic and Black Americans are more likely to develop Alzheimer's disease and dementia compared to non-Hispanic white Americans. They're greater familial risk for Alzheimer's disease, and they often have limited health care access, and thus Alzheimer's disease patients are identified at later stages of the disease and have poor treatment outcomes. There are also well-known sex differences in Alzheimer's. There are differences in prevalence, with women more frequently affected than men. Reasons for this increased prevalence in women are unclear, but with one possible contributor being that women live longer on average. There are differences in clinical and pathological manifestations between men and women. For instance, progression and pathology of mild cognitive impairment appears to differ between men and women, and there are genetic differences. In particular, the well-known gene APOE, risk gene APOE, has been found to have different effects between men and women. However, it's the only gene that has been looked at thoroughly for sex differences. Traditionally, most studies have combined men and women and not considered the differences or similarities that exist between males and females. And we're trying to, studies now are starting to look at that a lot more. Talk a little bit about that. So what have we learned in terms of genetics of Alzheimer's? Here I have a timeline. I put the names of genes that have been associated with Alzheimer's. And sometimes this is just the closest gene to a signal on a locus, because we really don't know what gene is critical, the causal gene. I've split these between genes that have risk variants that are rare, so less than 1% minor allele frequency in the population, or common in the population. And we see up until recently, we really hadn't associated many risk genes to Alzheimer's, at least with high confidence. In 2019, myself and a team of nearly 500 other researchers linked 5 genes to Alzheimer's risk. This was in addition to 19 previously identified risk genes. And you can see them listed here in the box on the left, with genes conferring larger risk and larger font. Figure on the right is the Manhattan plot of these genes, with known risk genes in blue and novel genes we identified in red. And for those that may not be familiar with what this plot is, essentially we're looking at associations with Alzheimer's risk variants across the genome. Stronger risk loci are higher on the plot. And so this was the largest ever genetic study at the time. And importantly though, it was only conducted in European ancestry populations. We also did pathway analysis of these data. We combined genes with similar biological profiles into pathways. We implicated 4 major types of biological processes, including several lipid cholesterol transport pathways, immunity, amyloid processing, and tau binding. We also saw that while the specific pathways are not enriched significantly for rare variants, if we looked across risk genes and pathways together, the AD risk genes and pathways are enriched for rare variants. This hints that there are other additional rare variants to be discovered, and that our efforts at whole genome sequencing, which gives us a deeper look at all the variations in the genome, should pay off. We also, so we can add these genes to the timeline, and some of the sequencing we have done, in fact, has started to find more genes and gene variants associated with Alzheimer's. Here we can see some additional genes that have been associated through the Alzheimer's disease sequencing project, which I'm a part of. It's a National Institute on Aging effort, and it's aimed at sequencing thousands of genomes in order to fully understand genetic architecture of disease. Importantly, all this data is made available to qualified investigators through NIAGADS, and I'll talk a little bit more about some of the data that's going into this the NIAGADS database in terms of especially increasing the diversity of the genomes that we have sequenced a little bit later. So how much of the heritability have we explained to date? Well, it looks like a bit over 50%. So we are still not accounting for nearly all the genes involved in disease. You can see the large green unknown area here in the figure. Most importantly, the findings here are really, for the most part, from European ancestry populations. So we essentially don't know what we don't know for other populations. It's a huge blind spot in our research. In 2013, though, the first GWAS in African Americans was conducted, led by Dr. Christian Reitz at Columbia as part of the Alzheimer's Disease Genetics Consortium. This study found that the gene ABCA7, gene first identified in European populations, was also associated in Black Americans. But you can see the sample size here was only 6,000 participants, roughly. So in 2020, we expanded this data set to include approximately 9,000 individuals, and we used the African Genome Resource Panel for imputation, resulting in over 35 million variants. This is an important point because having imputation panels that are kind of population specific really, I think, is going to be important moving forward for identifying variants that are associated with different ancestries. While still underpowered to identify many of the rare variants, it did increase the resolution of the analysis. And we identified several novel loci associated with Alzheimer's disease in this study. You can see here in red, in addition to several previous loci, including APOE and ABCA7, which we see in a non-Hispanic white population analysis. But I think what's interesting is that while we see most of the genes in non-Hispanic white studies, if you take a look, we don't see most of those genes that we see in non-Hispanic white studies. If you take a look at the pathways implicated, they strongly support the notion that immunity and lipid processing and intercellular trafficking pathways that underlying Alzheimer's in Black Americans overlap with the observed pathways that we see in non-Hispanic white populations. A new pathway emerging from our analysis is the renal system, suggesting a novel mechanism for Alzheimer's disease that needs further exploration. Kidney problems may be diagnosed at a later stage in Black Americans, which could exacerbate downstream consequences for them, including Alzheimer's disease. The kidney connection exemplifies how I think genetics, comorbidities, and healthcare disparities might converge to exacerbate Alzheimer's disease risk. But again, we need more research into why this pathway is associated. Overall, what this tells us is that While the major pathways involved in Alzheimer's etiology in Black Americans are largely similar to those in non-Hispanic white populations, many of the disease-associated loci within these pathways are different. Previous population studies have also shown that differences in genetic ancestry can have strong effects on gene expression. I can see two studies here. Genetic variants associated with AD are likely to exert their effect by regulating gene expression. And here in these studies, they examined primarily African ancestry and Alzheimer's disease, including a study on gene expression, which looked at African Americans and non-Hispanic whites, and one looking at RNA editing in these same populations. And they also found common biological processes associated with Alzheimer's disease, such as immune response and endocytosis, are present across populations. But again, much of these processes appear to be driven by distinct genes within the populations. Again, suggesting underlying common mechanisms, but driven possibly by distinct genes in diverse populations. Beyond this work, there are accumulating studies in individuals with primarily African, Asian, or Hispanic ancestry that also supports this conclusion, that there are both similarities and differences in genetic risk across ancestry groups, though I will note that these results are from small studies. So in fact, if we look at the sizes of the largest Alzheimer's gene studies to date, you can see that Black, Hispanic, and Asian Americans are substantially underrepresented in our studies. Largest GWAS in African ancestry, which we published in 2020, which I showed previously, had just 9,000 individuals compared to the 93,000 individuals in European ancestry that we looked at previously. And largest GWAS in populations from Hispanic and Asian ancestries are even smaller in size. So we really do have a power limitation in these other ancestries. This lack of diversity is not just an issue with Alzheimer's disease genetic studies. This figure shows that across the globe, we are missing a lot of ancestral diversity in our genetic studies of complex disease. It's a fact that could be important, I think, for combating Alzheimer's as well, since there are other complex disease comorbidities, such as cardiovascular disease that contribute risk to Alzheimer's, oftentimes disproportionately in underserved populations. So getting back to the ADSP, which I mentioned earlier, and again, all this data is available to researchers through NIGADS once it's produced. There's ongoing efforts to increase ancestral diversity in Alzheimer's genetic studies. A major one is supported by NIA, and it's the ADSP follow-up study, which is a global effort to generate whole genome sequencing on diverse genomes on the pathway to drug discovery. Through these efforts, we hope to identify new genes contributing to risk and protection of against Alzheimer's. We are examining in the multi-ancestral populations to identify the risk factors. You can see here, the goal is to have 37,000 individuals from each of the primary ancestral population groups, the largest 4 in the US. So we have 18,500 cases, 18,500 controls in each group. I'll just show you, the cohorts we're collecting towards this effort. They are from existing studies of Alzheimer's, dementia, or aging-related conditions. As long as they have cognitive data and we can make an Alzheimer's disease diagnosis, they are qualified for the study. And you can see a list of the 32 cohorts being sequenced here. I'll just note that the population diversity, it exists within the major ancestry groups as well. For instance, there are many Hispanic or Latino populations, including Mexican, Peruvian, and several Caribbean populations. There's an African study from Ibadan to complement the Black US data sets. There are genetic isolate populations such as Faroe Islands and Amish populations within our non-Hispanic white groups. And additionally, there are several large studies from foreign cohorts that will have their sequencing as part of this data set, and they include cohorts from India, Korea, Australia, and Portugal. So here you can see in terms of the NIA's goal of producing sequencing on 18,500 cases and controls on the 4 major ancestry groups. We are just about there for European ancestry, but we're falling short in the other ancestry groups. And I'll touch a little bit at the end about some of the work happening to make up for these differences. And I haven't really talked too much about sex differences in genetics yet, so to touch a little bit on that. We're starting to look at genome-wide data. A lot of researchers are, that's available. In our own studies, we identified several loci shared between sexes, indicated in the Manhattan plot in blue, but also several novel loci that are different between sexes in red. And several of these sex-specific loci had nominal association for interaction effects when we looked at them. In terms of other types of analyses, we're also looking at expression and methylation differences between males and females. Here are 4 recent epigenome-wide association studies, a total of more than 1,000 prefrontal cortex brain samples from Roxmap, Mount Sinai, London, and the Gasparini study. For each cohort, we employed 2 complementary analytical strategies: a sex stratified analysis that examined methylation Brock stage associations in males and females separately, and then one that looked at the same type of analysis in interactions analysis to look at the magnitude of the differences between males and females. And our analysis found 14 novel CPGs mapped to genes you see here. The circle genes are novel loci that are not significant in sex-combined loci, sex-combined analysis. And importantly, we were able to show that some of the genes known to be involved in Alzheimer's neuropathology, such as MBP and ZFPM1, appear to be driven predominantly by one sex. So why are these differences we see in genetics across populations and between males and females important? I think we can conclude that the study of primarily one population and a lack of consideration of sex differences leads to a lack of understanding of the complete genetic architecture of Alzheimer's and will intensify health inequalities. Importantly, translation of the research outcomes based on just one population to clinical practice can be dangerous, and it has implications not only for risk prediction, but for future therapeutic developments as well. I wanted to just kind of use APOE as an example to illustrate how this could work for precision medicine across populations. So APOE, one of the most important risk genes in Alzheimer's, we can see here the risk it confers varies greatly across populations, with East Asians having the highest risk versus Yorubans. It's an African population having the lowest risk. What is the cause of this risk effect difference in APOE? Is it due to environmental, ethnic related, or cultural factors related to diet or lifestyle? Or is it due to population specific genetic variations around APOE or other regulatory effects that are different between populations? I think before we answer that, just wanted to kind of be a little more specific about what we mean when we talk about race and ancestry to make sure we're all on the same page. Race itself is not biological. Ancestry is biological, and it's about history of genetic variation. We had populations living in isolation within geographic regions, and these populations today, particularly in the Americas, are admixed with individuals of European, African, Amerindian, and Asian ancestry. And if we look at a couple examples here, so these are all local ancestry plots in Black American individuals. Each panel shows the result of a select individual from each of the populations, shows 2 haplotypes for each chromosome. And we can see the wide range of ancestry percentages for each individual who is considered to be African American or Black as their racial category. So for instance, on the top left, individual is 99% African ancestry, whereas on the bottom right, this individual has 93% European ancestry and just 7% African ancestry, despite all of these individuals identifying by race as Black or African American. Some additional examples of local ancestry in Hispanic or Latino populations. You can see the, from 3 different source populations. Colombian individual has many more long European tracks. Cuban, Dominican Republic individuals have a lot more African this individual has a lot more African segments. Haitian individuals almost entirely made up of African tracks with some European regions, and the person from Puerto Rico is a mix and looks similar to Colombian, but with more African segments. So there's several things we can do with these local ancestry measures. One is to look at subcontinental ancestry by stratifying the genomes into only those pieces from a specific continent. We can look at all African segments or all European segments, for instance. How can this help with our previous question on why APOE risk differs across populations? In 2018, Rajabali and colleagues demonstrated that the reason for the difference in population risk for APOE carriers was due to surrounding inherited local genomic ancestry in APOE4 region. So for instance, if you have an African inherited APOE variant, your risk of Alzheimer's is less than if your APOE variant was from a European ancestral background. And your risk is particularly increased if your APOE4 variant has an Amerindian or Asian background. So knowing this, we can then look for specific regulatory variants, as I mentioned before, that may be influencing this risk in APOE4 across populations, and this could inform more effective therapies. So what are the clinical implications for this type of finding? Well, Alzheimer's disease starts in midlife, and with APOE4 carriers much earlier than others. This suggests that preventative measures in younger adults could lower Alzheimer's risk. We know that the risk of Alzheimer's disease for APOE4 carriers is different for patients from different populations. And now we know that the ancestry from which the patient inherited their APOE4 allele determines their risk for Alzheimer's from APOE4. And we can thus advise patients more precisely about their risk of Alzheimer's if we know the information about their ancestry at this location in the genome. We can also potentially develop and prescribe treatments informed by this information. So knowing a little bit more about genomic architecture based on ancestry and sex can also help us with risk prediction. I'm sure many of you have seen a risk prediction bell curve before on the left. This estimates an individual's genetic predisposition for a trait or disease. This predisposition is essentially a single number, and we can, based on the relative contribution of many variants across the genome, risk variants or protective variants, define a person's risk. So basically a way to take all the risk genes we identified and categorize them in this risk. In the top figure, we can see several key uses of these risk scores and throughout how we can use it throughout a person's life. And then in the bottom figure, you can see why we need to gather a lot more genomic information on diverse populations in order to make these risk scores more informative. This here is showing that using European predictions and across the genome, that our predictive ability is using European population risk variance is particularly lower for African and East Asian descent individuals. So I think we should get better risk prediction by using males and female specific genetic architectures also. So that's something else to consider. I think including showed data on differences in methylation. We have differences in expression across ancestries that also can be incorporated into these risk scores and see if they can also increase predictability. So what do we need to move forward? Well, I know this workshop is about existing data, using existing data, but we also know, as I pointed out earlier, that we need more participants from underrepresented groups in genetic studies. And so, you know, we are gathering more data. I'll get into that in a second. And again, all this data eventually will make its way into NIGADs and be available as existing data for other researchers. How can we gather this data in underrepresented groups? I think one thing to certainly consider is, why does underrepresentation exist in the first place? It's important to recognize that there are historical reasons for distrust in the scientific community among Black Americans especially. But I think it's also important to acknowledge that until recently, the funding agencies and much of the research community has not really focused and put in the amount of effort into partnering with underrepresented communities. Importantly, as I pointed out, with efforts like the follow-up study, the ADSP follow-up study, this is changing. But for many historical reasons, we see in surveys that there is widespread mistrust and belief that an Alzheimer's disease cure won't be shared equally, regardless of race, color, or ethnicity, especially among Black Americans. Surveys also show that there's a widespread racial or ethnic discrimination, especially among Black Americans seeking healthcare. And so we obviously, as a solution, I think, need culturally aware outreach efforts. There was a great lessons learned review by Drs. Bird and Parachak Vance that covers some of the successful approaches that can be implemented to overcome barriers that discourage minoritized groups from participating in research. And they include recruitment based on collaborative, mutually beneficial relationships between the community and research team. So thinking of the community as partners rather than research subjects. Employing team members with ties to the community and staff that reflect the community. Identifying key contacts for networking, including culturally sensitive community education that empowers people, such as information on Alzheimer's disease and lifestyle recommendations to promote brain health. And finally, providing ancillary services and incentives for participants, many of whom have other chronic disease conditions beyond AD that can help sustain longer-term relationships for longitudinal studies. Here is one of the efforts I'm a part of that aims to implement these culturally aware strategies in order to increase the number of individuals with Hispanic and African ancestry into genetic studies. It's the Recruitment and Retention for Alzheimer's Disease Diversity Genetics Cohorts in the ADSP. And our efforts include an initial goal for a recruitment of 4,000 Black Americans, 4,000 Hispanic Americans, 5,000 individuals from Africa across 10 different sites. All of this data will end up in the Alzheimer's disease sequencing project study. We hope. We intend to collect DNA, RNA, plasma biomarkers, and cardiovascular risk markers, along with extensive phenotype information on participants. We also aim to perform whole genome sequencing on these individuals. And importantly, we'll collect survey information on social determinants of health, which can also influence Alzheimer's disease risk. This effort is led by, you can see here, Dr. Sexis and Akanyami from Ibadan. And you can see in the graphic what these social determinants entail, including information on access to and quality of education and healthcare access, economic stability, and social and neighborhood indicators. Why is the social determinants important? Well, this could be critical to eliminating the health disparities. We see it in Alzheimer's among minority communities. In the graphic on the right, you can see the list of major risk factors such as less education, and here it appears, I mentioned quality matters, increased hypertension, and other lifestyle factors that can contribute to the risk of Alzheimer's over a person's lifetime. In addition,
excuse me, Dr. Kunkle, about 5 minutes or maybe a little bit less.
Got it, thanks. And On the right, you've seen recently a number of studies supporting the idea that living in a disadvantaged community can increase your risk of Alzheimer's disease. And so we hope by collecting information on social determinants of health, along with the genetic data, populations with increased risk of Alzheimer's, we'll be able to look at social determinants, genetics, and also gene-environment interactions. And this will help eventually in reduction of health disparities in disadvantaged communities. Just like to highlight another effort that is focusing on recruitment, and this is for Asian American and Canadians into the ADSP. Also, this basically mirrors the READ ADSP effort in study design, and is being led by one of our frequent collaborators, Dr. Lisan Wang at University of Pennsylvania. So to summarize, genetic risk factors for AD can be different depending on your ancestral background and sex. The mechanisms between ancestries appear to be similar, but some pathways may be important in some ancestries more than others. Investigating the relationship between genetic ancestry, sex, social determinants, and disease may inform precision medicine initiatives, risk assessment, and development of ancestry and sex-specific therapeutic and preventative strategies. And there's a significant and critical need to significantly increase diversity in our genomic analyses to truly achieve the ability to conduct cross-ancestry comparisons and clarify differences in genetic etiology and potential molecular therapeutic targets. So as I mentioned, the lack of information really is an issue, especially with power and being able to make clear and informed research discoveries. So with that, I'd like to thank you and thank all collaborators and especially the Alzheimer's Association and the NIA.
Okay, that was very, very informative. So we thank you for that presentation. I looked at least at the Q&A, and I didn't see any written questions. So just in the interest of time, I think we'll go ahead and move on to the next speaker and then gather questions in the last 15 minutes, 12 to 12:15. We'll do that.